Welcome to Aquatic Biology. Today, we are diving into one of the most fascinating and vibrant ecosystems on our planet, coral reefs. Often referred to as the rainforests of the sea, these underwater worlds are bustling with life, color, and complex biological interactions. We will explore how they are built, the delicate chemistry that sustains them, and why they are absolutely critical to the health of our oceans. Let us begin our journey beneath the waves. To truly understand a coral reef, imagine walking into a bustling, vibrant city like New York or Tokyo, but completely underwater. The corals themselves are the master architects and builders of this city. Over thousands of years, they slowly construct massive limestone skyscrapers. Meanwhile, thousands of species of fish, crabs, and sea turtles act as the citizens, darting in and out of the architecture. They rely entirely on these structures for food, shelter, and a safe place to raise their young. Without the corals tirelessly building and maintaining the city, this vibrant community of marine life would have nowhere to live, leaving the ocean floor as a barren, empty desert. Now that we have a picture of this underwater city, let us meet the builders. In formal biological terms, a coral reef is a complex marine ecosystem built by colonies of tiny animals called coral polyps, which belong to the phylum Cnidaria. These polyps secrete hard calcium carbonate skeletons that accumulate over millennia. But they do not do this alone. The secret to their incredible building power lies in a mutualistic symbiotic relationship with microscopic algae called zooxanthellae, which live right inside the coral's tissues. The algae perform photosynthesis, providing the coral with up to 90% of its energy needs. In return, the coral provides the algae with a protected environment and essential nutrients. It is a perfect partnership. Because of this vital partnership we just discussed, corals are very particular about where they live. The microscopic algae living inside them absolutely requires sunlight to photosynthesize and produce food. Therefore, coral reefs cannot grow in the deep, dark abyss. They are typically restricted to shallow, crystal-clear oceanic waters where light can easily penetrate. Furthermore, they thrive in warm temperatures and surprisingly nutrient-poor environments. If the water has too many nutrients, other fast-growing organisms like seaweeds can take over and block the sunlight. So this delicate balance of clear, warm, and shallow water is the strict environmental requirement for our underwater megacities to flourish. Understanding where they grow leads us to an important question. Why should we care about them? Coral reefs are often called the rainforests of the sea for a very good reason. Despite covering less than 1% of the ocean floor, they support roughly 25% of all marine species. Their biodiversity is staggering. Beyond biology, they act as massive natural breakwaters. They absorb wave energy, protecting coastal human communities from devastating storms, hurricanes, and erosion. Economically, they are absolute powerhouses, supporting the livelihoods of millions of people through fisheries, tourism, and even the discovery of new life-saving medicines. If reefs disappear, coastal economies collapse, and a quarter of marine life loses its home. Knowing how vital these structures are, let us look at how they are actually constructed at a chemical level. To build their massive limestone cities, corals extract dissolved minerals from the seawater surrounding them. Specifically, they build their skeletons using a form of calcium carbonate called aragonite. The ability of a coral to extract these building blocks depends entirely on a chemical property known as the aragonite saturation state. If the water has plenty of these building blocks, corals can build easily and rapidly. However, if the ocean becomes too acidic, these building blocks become scarce. Under highly acidic conditions, the reef cannot grow, and the existing limestone structures can actually start to dissolve back into the ocean. This chemical relationship we just discussed can be expressed mathematically using the aragonite saturation state formula. Let us break down the symbols. The Greek letter omega represents the aragonite saturation state, which tells us how easy it is for corals to build. In the numerator, we multiply the concentration of calcium ions by the concentration of carbonate ions. These are the raw building blocks. The denominator, Ksp, is the stoichiometric solubility product, a constant that depends on water temperature, salinity, and pressure. In plain English, this formula compares the actual amount of skeleton building materials in the water to the minimum amount needed. When omega is greater than 1, the water is supersaturated, and corals build easily. If omega drops below 1, the water is corrosive, and skeletons dissolve. Now that we have our formula, let us see this with a concrete example. Imagine you are a marine biologist testing a newly discovered reef. You measure the calcium ion concentration as 0.010 moles per kilogram and the carbonate ion concentration as 0.0002. The solubility constant for this specific water is 0.000001. We substitute these values into our formula. First, we multiply the two concentrations in the numerator to find the total available building materials, giving us 0.0000002. Finally, we divide this by our constant. The result is an omega value of 2.0. Because 2.0 is significantly greater than 1, the water is supersaturated, meaning this reef is in a healthy chemical environment and actively growing. We've just seen how chemistry dictates reef growth, but chemistry is not the only limiting factor. As we learned earlier, corals rely heavily on their symbiotic algae for food. Because these algae perform photosynthesis, they absolutely need sunlight to survive. Every species of coral requires a specific minimum percentage of surface sunlight to maintain enough energy production. However, as you go deeper into the ocean, the water absorbs and scatters the light. This means the deeper you go, the darker it gets. Eventually, you reach a depth where the light is simply too dim for the algae to photosynthesize, and the coral would starve. This relationship between depth and light is crucial for predicting where reefs can survive. To calculate exactly how much light reaches a certain depth, we use the Beer-Lambert law. Let us look at the variables. I sub z represents the light intensity remaining at our target depth z. I sub 0 is the initial light intensity, right at the ocean surface. The letter e is the base of the natural logarithm, which models exponential decay. The variable k is the light attenuation coefficient, which is a measure of how murky or clear the water is. A higher k means murkier water that blocks light faster. Finally, z is the depth in meters. Physically, this formula tells us that as depth z increases, or as the water becomes murkier with a higher k, the light available to the corals drops off exponentially. Let us apply this law to find the maximum depth a coral can survive. Suppose a coral requires at least 10% of surface light, so I sub z equals 0.10 times I sub 0. The water's murkiness, K, is 0.15. We set up our equation. Notice that I sub 0 is on both sides, representing the initial surface light. We can divide it out, leaving 0.10 equals e to the power of negative 0.15z. To get our variable z out of the exponent, we take the natural logarithm of both sides. The natural log of 0.10 is approximately negative 2.30. Finally, we divide by negative 0.15. The negatives cancel, giving a maximum depth of 15.33 meters. Below this depth, the coral would starve. Now that we have calculated the limits of depth and chemistry, let us look at how a reef is physically structured in the ocean. This diagram shows a cross-section of a typical reef ecosystem. Starting on the left, we have the shore. Moving outward, we enter the shallow reef flat, a protected area where sunlight easily penetrates the clear water, as shown by the yellow arrows. Next is the reef crest, the highest point of the reef that takes the direct impact of ocean waves, acting as that natural breakwater we discussed. Finally, the structure drops off sharply down the reef slope into the deep, open ocean. Notice how the sunlight arrows fade as the water gets deeper on the slope, perfectly illustrating why corals cannot grow in the dark depths. We have covered a lot of ground today, so let us review our key takeaways. First, coral reefs are magnificent ecosystems built by tiny polyps working in perfect harmony with photosynthetic algae. Second, they are incredibly sensitive, requiring specific environmental conditions like warm, clear, and shallow water. Third, we learned that the aragonite saturation state dictates whether corals can build their limestone skeletons or if they will dissolve. Fourth, the Beer-Lambert law shows us exactly how light attenuation limits the depth at which these corals can survive. Understanding the mathematics and chemistry of their environment helps us predict how these delicate rainforests of the sea will respond to a changing world. Protecting them is essential for preserving marine life and our coastlines.